So, good morning, everybody. Can you hear me?
Yes.
Yes.
Thank you. So I understood there's an exam today, so people might be late or will watch then the stream. Nevertheless, we will start. I welcome you to this teaching session on sample preparation and subcellular fractionation. What I would like you to do is that you please sign up with your name and matricle numbers in the chat for registration. That we will need that later. We will start now, and we make several breaks in between for grabbing a coffee. And first, I would like to introduce myself to you. So my name is Lucas Huber. I'm a medical doctor and cell biologist working at the Biocenter at Innsbruck Medical University. So I'm a signal transduction cell biologist. I work with signaling from lysosomes, from endosomes. I work with rare diseases and with cancer. And I will give you today a kind of practical course how you can use subcellular fractionation in your further work on the bench, what you can learn from it, what you can expect from it. We will go for protocols. You will see videos that will demonstrate the procedures I'm going to introduce you today. And you will get the homework afterwards. Now, for the ones who are not in the live stream in Zoom, now this is no problem. The ones who have registered, they can, excuse me, they can watch the lecture afterwards also on my YouTube teaching channel, where you find also all kind of different cell biology lectures. Now, do you have any questions right now? If you don't, I would like to encourage you that you ask me questions during the lecture, that you turn on your mic and interrupt me, or that you write a question in the chat, and I will try to answer all of them online as good and efficient as possible. Now, a few words to the protein contents and to the human proteome. The human proteome is a huge entity, and that is the reason why we want to dissect it when we analyze it. When we follow the hypothesis of one gene is one protein, there should be at least 20,000, even more, non-modified canonical human proteins. Now, when we take into account alternatively spliced products, those containing single amino acid polymorphisms, and those that undergo post-translational modifications, and there are more of 200 chemical post-translational modifications out there, then you come to a number that as many as 100 different proteins can potentially be produced from a single gene. And that gives you immediately a dimension how huge the human proteome is. And it's very complex. So take a simple body fluid like blood plasma. This is a very complex mixture with a very dynamic range of protein concentrations. And they vary by more than 10 orders of magnitude. And at the same time, the protein concentration range of tissues or cell lines is within 7 orders of magnitude. And the challenge is then detecting low and ultra low abundant species with concentrations from less than to the minus 12 mole in the presence of high copied protein molecules, for example, at concentrations from more than 10 to minus 6. Now, unfortunately, many of the proteins, the signaling proteins we are most interested in, exist only in a few thousand copy numbers per cell. Let's think about the most popular oncogene that is mutated in more than 70% of all solid tumors, and that is RAS. You have a few thousand copy numbers of these small GTPase. And there's a reason why cells keep such important gateway proteins, molecular switches at low concentrations, because when you have too much of such proteins, then many signal transduction processes would be over amplified, for instance. Now, but these proteins are the ones that we want to find. And I will give you this picture of this iceberg. Because we need technologies that allow us to access intracellular organelles and multi-protein complexes. And usually these multi-protein complexes are associated with subcellular compartments. They are tethered to membranes of endosomes, lysosomes, of the Golgi, of the endoplasmic reticulum, of other vesicles, vesicular carriers in the cell. They are docked from inside to the plasma membrane, or they are in the cytoplasmic space and they are transported along microtubules or actin filaments. And therefore, subcellular fractionation is a very useful technology whenever you want to dig deeper into a cell and you want to find the tip of the iceberg underneath the water surface. It enriches these low abundant proteins and signaling complexes, and it reduces the sample complexity. And that is very important because proteomics techniques have problems with these huge concentration variations. The approach is flexible and adjustable. It can be very efficiently combined with functional analysis. Think about mitochondria. You can purify mitochondria even out of patient samples, and you can also test them functionally. You can test them molecularly. So there are many possibilities that arise from that, and it's combinable with more dimensional electrophoresis procedures, gel-independent techniques, and all mass spectrometry techniques that you will see later during this talk. However, subcellular fractionation remains a major bottleneck. And I just have to go back now for a moment. Give me just a second and launch the presentation in this format, that we are ready for the videos. So subcellular fractionation is still a bottleneck that is technically very demanding. What you see here is an electron micrograph of a plant cell. You see the cell wall, and you see in very nice high resolution different Golgi stacks. You see mitochondria, you see lysosomal structures, you see ER, you see endocytic vesicles, exocytic vesicles. They all look very different to you. However, they share similar physical properties, and they're extremely tensely packed within the space of a cell. And there are very remarkable differences in when you fractionate tissues or when you fractionate cultured cells. And we will come to that notion a bit later. Now, the biophysical differences from these organelles, and most of our purification techniques are based on biophysical differences. However, we will add also immunological differences and functional differences to the list of methods today. But the biophysical properties are given to these organelles through the proteins and the lipids they contain. And all the organelles that I'm going to show you in this next video, we can purify. The video you will see in a minute. Now, therefore, we will introduce these organelles. We'll talk about a certain membrane transport pathway that we take as a paradigm today for purifying different components out of that. And then we will go into the fractionation of organelles. The most and first attention will be given to the homogenization. How do we break up cells? Then we will devote our attention to organelle separation per se, density gradients, density shifts we will discuss, and immune isolations, fluorescence, activated organelle sortings, just to name a few for you. Now, look at this introduction.
Cells are the smallest living units of an organism. All cells have three things in common, no matter what type of cell they are. All cells have a cell membrane, which separates the inside of the cell from its environment, cytoplasm, which is a jelly-like fluid, and DNA, which is the cell's genetic material. There are two broad categories of cells. The first category is eukaryotic cells. They have organelles, which include the nucleus and other special parts. Eukaryotic cells are more advanced, complex cells, such as those found in plants and animals. The second category is prokaryotic cells. They don't have a nucleus or membrane-enclosed organelles. They do have genetic material, but it's not contained within a nucleus. Prokaryotic cells are always one-celled or unicellular organisms, such as bacteria. So, what are organelles? Organelle means little organ. Organelles are the specialized parts of a cell that have unique jobs to perform. Let's start with the nucleus, the control center of the cell. The nucleus contains DNA, or genetic material. DNA dictates what the cell is going to do and how it's going to do it. Chromatin is the tangled, spread-out form of DNA found inside the nuclear membrane. When a cell is ready to divide, DNA condenses into structures known as chromosomes. The nucleus also contains a nucleolus, which is a structure where ribosomes are made. After ribosomes leave the nucleus, they will have the important job of synthesizing or making proteins. Outside the nucleus, the ribosomes and the rest of the organelles float around in cytoplasm, which is the jelly-like substance. Ribosomes may wander freely within the cytoplasm or attach to the endoplasmic reticulum, sometimes abbreviated as ER. There are two types of ER. Rough ER has ribosomes attached to it, and smooth ER doesn't have ribosomes attached to it. The endoplasmic reticulum is a membrane-enclosed passageway for transporting materials, such as the proteins synthesized by ribosomes. Proteins and other materials emerge from the endoplasmic reticulum in small vesicles, where the Golgi apparatus, sometimes called the Golgi body, receives them. As proteins move through the Golgi body, they're customized into forms that the cell can use. The Golgi body does this by folding the proteins into usable shapes, or adding other materials onto them, such as lipids or carbohydrates. Vacuoles are sac-like structures that store different materials. Here, in this plant cell, the central vacuole stores water. Going back to the animal cell, you will see an organelle called a lysosome. Lysosomes are the garbage collectors that take in damaged or worn-out cell parts. They are filled with enzymes that break down this cellular debris. The mitochondrion is an organelle that is the powerhouse for both animal and plant cells. During a process called cellular respiration, the mitochondria make ATP molecules that provide the energy for all of the cell's activities. Cells that need more energy have more mitochondria. Meanwhile, the cell maintains its shape through a cytoskeleton. The cytoskeleton includes the thread-like microfilaments, which are made of protein, and microtubules, which are thin, hollow tubes. Some organisms, such as plants, that are photoautotrophic, meaning they capture sunlight for energy, have cells with an organelle called a chloroplast. The chloroplast is where photosynthesis happens. It's green because it has a green pigment called chlorophyll. Plant cells also have a cell wall outside of their cell membranes that shape, support, and protect the plant cell. Animal cells never have a cell wall. There are many other unique structures that only some cells have. Here are just a few. In humans, for example, the respiratory tract is lined with cells that have cilia. These are microscopic hair-like projections that can move in waves. This feature helps trap inhaled particles in the air and expels them when you cough. Another unique feature in some cells is flagella. Some bacteria have flagella. A flagellum is like a little tail that can help a cell move or propel itself. The only human cell that has a flagellum is a sperm cell. In summary, remember, eukaryotic cells are plant and animal cells with a nucleus and membrane-enclosed organelles, while prokaryotic cells are unicellular organisms without these things. All cells have a cell membrane, cytoplasm, and genetic material. And even though only plant cells have chloroplasts, both plant and animal cells have mitochondria.
Yo, and the message is that all those organelles, cell parts, you can purify. You can purify the membrane enclosed organelles, you can purify ribosomes, proteasomes, you can purify flagella, cilia bodies, you can purify and enrich the cytoskeleton, you can purify plasma membrane, you can dissect the nucleus, and you can purify the chromatin. So that is all based, provides the basis for our molecular understanding how cells work. And these subcellular fractionation techniques, they have driven our understanding of modern cell biology. And what you see here now is this very schematic representation of the different transport routes, for instance, where you have the outgoing route, the biosynthetic pathway, as we call it, where proteins, for example, that have to reach the plasma membrane, membrane proteins, or also cargo proteins that have to be released outside the cell, are synthesized on ribosomes in the endoplasmic reticulum, where vesicles part off and transport them to the Golgi, where they enter the Golgi on the cis side, passage through the middle part of the Golgi, the median cisterne, reach the transcisterne, and then are transported to the trans-Golgi network. In the Golgi, they are modified biochemically. Sugars are added, are trimmed, phosphorylations are added, And then they reach the kind of post office station, the TGN, where they are packed into different vesicles and are transported either to the plasma membrane or with certain codes also to the endosomal system. Vice versa, we have the incoming protein transport pathway, which we call endocytosis. And in endocytosis, you should keep in mind for the further understanding that different compartments pre-exist in the cell. These are the early endosomes, these are the multivesicular bodies, and these are the late endosomes, and finally the lysosomes. Important it is also the role of the cytoskeleton, because all these transport steps are facilitated through the help of the cytoskeleton. Here only the microtubule organizing center is shown, but also the actin cytoskeleton contributes to that. And we will, for our further understanding of how subcellular fractionation work, for didactic reasons, remain with the endocytic pathway. But in principle, everything I'm going to show you today is applicable for many other organelles as well. And you will understand later why. What my goal is to show you how you can purify such organelles, that you understand the protocols that are out there, to supply you an addition with protocols that you could adapt that then to your needs. When you work on mitochondria or you work on a protein that shuttles between the cytoplasmic compartment and the membrane compartment, and your supervisor wants you to show that. When you work with nuclei, when you work with proteins that should remain in the nucleus or leave the nucleus, when you work with receptor settings on the plasma membrane or in the synapse, all that you can use, and we will exercise that today. So what are the key points of cell fractionation? The first point and the most important is the breaking open of the cells and the separation. The separation then of this gemisch, so to say, of the parts into pure fractions. And this requires usually a considerable high number, a large number of cells. The breaking open of cells requires the lysis and the homogenization. And this cell disruption can be achieved chemically, for instance, for alkaline reagents, for example, or organic solvents. But this is pretty harsh. You can use detergents that solubilize membranes. You can use enzymes, chitinase or lysozymes that digest the cell wall away. And keep in mind, all these methods you can use for plant cells as well as for human cells, for animal cells. They are all uniformly usable. We can also use physical methods like osmotic shock or freeze and thawing of cells, mechanical methods like ultrasound sonication, homogenization with homogenizers of different kind, I will show you. You can use a French press, I will show you what that is, or you can simply use a syringe with a needle. And one of my goals is to use methods as simple as possible that I'm teaching to you, where you have reagents, instruments that you basically find in every lab. Like the last example, a syringe with a needle. It is as simple as that. You just need the right buffers, the right conditions, and then you can do that independently from where you work, in which lab you work, and how the lab is equipped. Let's come to the chemical disruptions. So for these detergents, such as Triton X100 or MP40, have the unique feature that they permeabilize cells by solubilizing membranes or parts of the membrane. And you have to use mild detergents at the right concentration to make holes in the plasma membrane. And that is symbolized here in this picture, where you see that the detergent has solubilized parts of the membrane, and then the cytoplasmic content, plus the vesicles, plus the mitochondria, plus everything can leak out, and you get access to it, and you can further purify it. The disadvantage is that detergents can be expensive. The good detergents are very expensive. They can denature proteins, and at the end of the day, you want to analyze the proteins as native as possible. And they have to be removed after the disruption because all kind of downstream analytical methods like mass spectrometry or functional measurements would be disturbed by the presence of detergents. So the concept is simple, but when you look more carefully at it, it has its drawbacks. What is a French press? Now, a French press is basically a stainless container, stainless steel container, where the cells are placed in it. That you can see here on the figure on the right. And there is a little hole, a small hole at the bottom of that container. And then you have a piston, and with large pressure that you apply, you basically press the content through that hole, and therefore it's called French press. It's a bit like comparable to orange juice presser. You know this Italian designed metal orange juice pressers where you put half of the orange on it and you press a piston down, and then the juice is released. And this is something we usually use for breaking up bacteria when we use bacteria as protein factories, for instance. But you can also use it for cells. But as you can imagine from the simplicity, it is very difficult to apply, that it is not too harsh. Because when it's too harsh, many things can happen that we don't want to happen. We will talk about that soon. Then we have a Sonicator. A Sonicator can, with its tip, be directly immersed into a cell suspension. And that is shown here. You see the tip of a Sonicator, and you see the remaining parts of a cell. They looked before like this, and now you put in the Sonicator, they are disrupted through the vibrations of a high frequency sound. And these waves disrupt the cells, and the cells then vesiculate. Like during mitosis, when many organelles vesiculate, but here also the plasma membrane vesiculates. Then we have homogenizers, and these homogenizers with downsers. So this is a closed vessel, as you can see here, and usually it is made out of glass. Then you have a plunger that fits tightly in. And you can buy these devices. They are rather cheap, the glass vessel plus the plunger, with different distances from the side of the plunger towards the wall of the glass vessel. And this distance can vary. They can manufacture it in different distances. And through this variation of the distance, these entry holes on the side become either wider or smaller. And you can adapt that to different cells, different cell sizes, or different cell shapes. We will also learn today that round cells are more difficult to homogenize, for instance, than floppy-like fibroblasts or epithelial cell sheets. And the cells are very similar in a very similar way, we have seen it before for the French press, disrupted as they pass between the plunger and the cell wall. Now, this is how these instruments look like. You can have a plunger, for instance, also with kind of a drilling device combined. You can have them in different shapes, different forms, in different sizes, and they are rather cheap. And what also is an advantage, you can cool these systems because you can put them in an ice bucket, and I will explain you why cooling is so important. And this is the device that I will recommend you and that you also find in our protocols. This is the homogenization with a syringe and a needle. And this is cheap, easy, and efficient. And the special syringe that you see here is a 1 milliliter syringe. We call them also tuberculin syringes. you can have these syringes in different sizes with different volumes. So that gives you already the idea why this device is so adjustable to your needs, because sometimes you have larger volumes, sometimes you have smaller volumes. And what you can also see is here is this color of the needle. So the needle are color coded, and that the entry opening of the needle is measured in gauge. And you have needles with 21 gauge, 22 gauge, 24 gauge. And from this gauge number, you can derive immediately the diameter of the needle. So the smaller the needle is, and that you know from the vaccination, the less it hurts, but the longer it takes to get the liquid through. The larger the needle is, for example, when you want to collect blood from the patient, from the arm, for instance, then you would like to have a larger needle, because then the flow is much faster and you do not have to torture the patient too long. For us, this has a very practical point of view, because here we can vary with the different needles the pressure we put on the cells, and that becomes important because cells have different sizes and different shapes. And the shear forces that you generate by pressing the cell suspension here through disrupts the cells. And then the organelles and the nuclei are released, and that the needle and the syringe can be varied and reused is also understandable. Now, we need some general remarks before we come to this homogenization per se. In certain cell types, we have to keep in mind the cell number is very instrumental, because the cell passage number, excuse me, is very instrumental because it influences the homogenization result. Use always cells with a known passage number. When cells are at too high passage number, they undergo aging, and cellular aging. And when cells age, they usually change their appearance, their morphology. They become very flattened out on the culture dishes. They rearrange their cytoskeleton, and they become pretty tough to homogenize. And you have to apply more shear forces. Now, when you apply too much of the shear forces during the procedure, then you can break organelles. And this is not only of disadvantage because you want, in principle, to study intact organelles and not broken organelles. It is also of disadvantage because you release nasty content out of organelles. Think about the lysosome. The lysosome is a bag full of enzymes. It contains hydrolases. It contains lipases. So it will, in the moment when you break lysosomes during the procedure, the content, the enzymatic content leaking out of the broken organelles will start destroying your sample. At the same time, think about the nucleus. When the nucleus is broken, what does it release? DNA. And from your practical work at the bench and the practical courses during your study, you know that when you purify DNA, it's a slimy clump. And this slimy clump would unspecifically generate aggregates of organelles, aggregates of cytoskeleton, aggregates of cell debris that is generated. So it's like a glue in the bottom of your tube that you would like to avoid. And therefore, it's always important that you record really carefully which passage numbers you have used in order to have reproducible results. And this is an important point for you to take with you. You have to write down everything into your lab notebook. When you harvested the passage, was it passage 26 or was it passage 46? Was it passage 7? How the cells looked like? And then, you will be able to trace back unreproducible results, because you will find out that on that day you have used basically 2 old cells, and therefore the yield and the efficiency were so low. What is also an important parameter to keep in mind with cells is the confluency. The confluency of the cells on the plate is a critical parameter because when the cells become confluent, they form cell-cell interactions. And these interactions can be junctional complexes, and they must be broken during the homogenization. And that is a very helpful part. And you will be surprised why. When you break cells that form an epithelial sheet, for example, or cell-cell junctions, the shear forces that you have to generate in order to break the individual cells are lower than the shear forces you have to generate when the cells are swimming roundish like lymphocytes in a suspension. The reason is that when you start applying shear force on one end of the cell sheet that you take off of the dish, these shear forces multiply during the procedure, and when you disrupt one cell, this disrupts the cell next to it and a cell next to it, and that means you need less pumps with your plunger, less pumps with your syringe, or less pressure with your French press. When you have round cells that are like lymphocytes or trypsinized cells, where the cytoplasma to nucleus ratio is completely different than to an epithelial cell or to a fibroblast. When you look at my hands now, when this is a round cell, the entire cell, like a lymphocyte, is basically filled with the nucleus, and the cytoplasmic rim is very, very small. Whereas other cells, like fibroblasts or epithelial cells, they have a much larger cytoplasmic space, and then they wobble a bit more, and that makes it easier to break them up. So therefore, you have to check if the cells have reached at least 80% confluency before you start your experiment. You should also take care how you put your plates into the tissue culture incubator. You make sure that when you trypsinize your cells and you seed them out for the experiment, that they are completely flat on the shelf in the incubator. Because once the dishes, for example, are tilted a bit, because you put them in a sloppy way, then the monolayer cannot be formed. They will collect all on one side, and they will not form monolayers, and they will not sit down properly on the plastic. So these are just simple and easy to follow recommendations. The next recommendation is that all steps have to be performed on ice. And this is like such a bench place. Looks like, this is an ice bucket. You can also have simple styrofoam containers. These are ice buckets. You fill them with crushed ice. You put a metal plate in that the cells nicely sit flat on this ice, and you cool them down. All buffers need to be cooled down that you use also when these are syringes, for example, or for the homogenization, or these plastic Pasteur pipettes, they need also to be cooled down. Why? The main reason is that all nasty enzymes that would disturb the quality of your sample act much slower or are inactivated at 4 degrees. These are proteases, these are phosphatases, these are lipases, hydrolases. When you cool down a cell, the entire metabolism is very much reduced. And this is very helpful for you because you prevent protein digestion or you prevent dephosphorylation of proteins or lipid digestion. So your sample needs to be in good shape. What you should also take into consideration is during the summertime, for instance, when you would have a lab where the window is directed into the southwest direction, like for example the Max Perutz laboratories are towards the Tangente, or the laboratories at the IMP or at the IMBA towards southwest. Then in the afternoon, they warm up due to the sun that comes in for the windows. And then it's already recommended that you perform these homogenizations of the cells in the cold room. So that is also one of the reasons why you see jackets hanging in front of the cold room, because scientists have some steps to perform in a cold environment in order to keep the sample quality up. Another reason is that you can use this temperature block to slow down all vesicular compartment traffic. That means you can introduce markers into a vesicular compartment by endocytosis or by genetically engineered exocytic markers, and you can block them at different steps of this transport, and you can enrich the markers in a certain subpopulation of vesicles that you want to purify. Why? Because the motor proteins are inactivated and because the entire ATP metabolism is slowed down and the cytoskeleton is disturbed. The microtubules, for instance, are completely disrupted. And when you raise the temperature again, internalization or vesicular transport proceeds, and the microtubules reform. So that's the reason why we perform all steps on ice. Now, for the homogenization per se, you take a confluent cell culture, put it at 4 degrees, like I have shown you on this metal plate, and you collect the cells first by scraping. And we are using these simple cell scrapers. They exist in versions out of metal with a little Teflon piece here, but they exist also in plastic sterile packed devices. Important is that this little extension here out of Teflon is rather soft and not too hard, because you want to scratch the cells in buffer here down, that they come off, that you can further proceed them, but you would not like to scratch open them already here. They should come off as intact as possible. And when this is an entire monolayer that you can scrape off, then they look like a little piece of skin that comes off when you had a sunburn, for instance. And when these cells are then coming off after a few days, these little pieces of skin. And that is exactly what it should look like. And then with this solution that you collect here, you start the homogenization. But don't worry, I will show you a video where everything I explained will be shown here once again. So keep in mind, the cells are released from the dish by scraping with a sharp edge of a rubber policeman, and the homogenization per se is easier when you have a relative high density of the cells. Yeah. Verify before you start the homogenization that you have a nice solution and no big aggregates are visible in the cell suspension. And it looks homogeneous. I will show you in the video then how it should look like, and this is important before you disrupt them with the homogenization. And the cells then per se, this what you have scraped off in buffer, are then homogenized by passage for a needle in our video that I will show you, or for the tip of a pipette. And that generates the first fraction, and that is a post-nuclear supernatant, because this is called PNS. You will disrupt the cells by homogenizing, and then you will make a first centrifugation step. And we use the method of differential centrifugation. I will introduce that to you a bit later today, in order to separate organelles based on their size, on their weight, and on their shape. And here you can spin out the nuclei, and then you have the homogenate. The post-nuclear supernatant reveals this homogenate that you take out of the tube, and then the bottom of the tube remains the nuclear pellet. Now, when you are working in a lab that is involved in transcription factor research, or in nuclear pore complexes, or that investigates the nuclear lamina or whatever, then you will proceed in these protocols with the nuclear fraction and will further clean it. Today, we will take the homogenate, and this homogenate will be the basis of purifying all the organelles out of it that are swimming in that homogenate. What is a good recommendation is to monitor this homogenization process under phase contrast microscopy. And for this, it's rather easy. You can use a microscope that is standing in the tissue culture lab, or a simple histology light microscope. You take a glass slide, put a drop of this homogenate on and the cover slip, and you go to the microscope. And then you look into the microscope, what do you see swimming there? So you see entire cells swimming, you see broken cells that are partially broken, you see many released nuclei swimming around, many little vesicles and organelles that float around. And then you look at that under the microscope, and when you have about 80% of the cells broken, then you should stop the procedure. Because when you overdo this homogenization, what will happen is that you break the nuclei, because the nuclei are then exposed to more and more of the shear forces. And when you see nuclei starting to break so that the DNA leaks out, and that is very easy to see because you see a disrupted nucleus where something is floating out, then you should stop the procedure. Then the homogenization is done, and then the next purification steps will take place. This shows you this homogenization with the needle. So what you have scraped off the dish, you put in a 15 milliliter tube, Falcon or Greiner or whatever you have. And then you have this syringe that you put in, and you see here we have a gray needle. Yeah. And then you put the syringe, it fits exactly in a way with that needle that the needle touches the bottom of the tube, and you can fix it here with 2 fingers, and then you pull basically up and down. And what is important here, during the pulling up and down, you should not generate air bubbles here. So why is that important, not to generate these air bubbles? Air bubbles would lead to unpredictable shear forces at the tip of this needle of the syringe. And then you cannot control the shear forces anymore, and cells might escape the shear forces, other ones might be hit too much. So avoid air bubbles when you put that up and down. In the protocols is written you need 5 strikes with a syringe with that size, or 3 strikes, and then you should go to the microscope and control the homogenization. But when you have seen that once, what I'm showing you here and what you will see in the videos, you will understand the lab protocols that are out there. Because otherwise, it will be pretty ununderstandable for you when you read you need 3 strikes of a syringe. But it is as simple as that. And with the number of the strikes of the syringe that you apply, you can at the same time direct the number of shear forces you apply. And when you have cells that require more harsh shear forces, you take a higher number. When you have cells that are more sensitive, you can stop after 1, 2, or 3 strokes already. Very often we have these aggregates in the sample during homogenization. These are cytoskeletal elements. You know, we don't forget we perform all of these steps on ice, and these cytoskeletal elements entrap the organelles. And these aggregates we need to resuspend, yeah, because when we apply centrifugation step afterwards, these huge aggregates would trap, for instance, smaller organelles, and when we gradient purify them later, they would go into the wrong fraction. That is what I said. When the cells are over homogenized, the organelles will break and release their content. And as a direct consequence, the cytosolic fraction will be contaminated with typical organelles bound material, and you lose organelles. The breakage of lysosomes, I have mentioned already, will release these nasty enzymes, and this nuclear DNA breakage releases DNA and acts as a glue somehow. So certain cells are harder to homogenize because they tend to be round up when they are scraped or trypsinized, and some cells are in suspension and are grown in suspension because they never adhere, like lymphocytes, for example. But there we have also methods like hypotonic shocks, so that we need to swell them before. Interestingly, it's much easier to fractionate organelles out of tissues due to the reasons that I explained to you before, that the shear forces multiply. And therefore, it's no surprise that the first protocols of purifying Golgi steps were established in the 70s, and by George Ballade, for example, Catherine Howell, and other pioneering cell biologists who also received the Nobel Prizes for that, for understanding what the Golgi, for example, is. And they used rat liver because that is a fairly large tissue source. It's a very active epithelial organ, and it has a very active Golgi, and they could purify Golgi 40 years back or so to homogeneity. And this was much more difficult than later for cell biologists to apply that to tissue culture cells due to the reasons that I have introduced to you. So what we are going to do now is we are looking at a video that I have filmed with one of my co-workers in the lab, where these procedures, as you see here from the protocol, this is now a video view on the protocol, what you will need and how these protocols look like. You see also this graphic looks familiar to you. This is, for example, the protocol that our students get for the practical course in the lab when during their studies they have their cell biology practical course 1, 2, or 3. And there the procedure is explained with the cell culture, the homogenization. There is an introduction. This is how these protocols look like. I will comment the video for you, and I will start the video now. So now you see the description of the protocol. Keep everything strictly on ice. You always get a list of equipment and material. You see here the ice bucket where this metal plate is launched. This is how the working place looks like. So now my co-worker goes to the tissue culture incubator, takes the cell dish out and places it on ice. What is written on then? These are HeLa cells. P26 means passage number 26 and the date when they are seeded. So everything is prepared here in this ice bucket, the buffers and reagents you need, the syringe, the scraper you see here. So the first thing is when the cells have cooled down, we exchange the medium to 10 milliliters of ice cold BBS, and we wash the cells. So the medium, the dish is taken, the medium is simply poured off into a beaker or a glass that you have on the side of the bucket. You take an electric pipette aid, add 10 milliliters of BBS, but again, be careful that you blow it in the side on the wall and not in the middle of the dish, because otherwise you will blow away the cells. So carefully release the medium, the BBS, the ice cold BBS in the dish, shake it several times and take it off, and then you go again and you repeat this procedure. So then he pours it off again. Then he adds 5 milliliters of this phosphate buffered saline, ice cold, and this will be the volume that we will require for scraping the cells. Now the cells are washed, medium is washed away, and now he fixes the scraper and rotates the dish. And then you see that pieces come off already. He tilts it, and then he scrapes after it to remove everything, and there are these little white pieces. They look like a bit of skin that comes off after a sunburn. And now you take that with a pipette, take that out, you take a plastic Pasteur pipette, which has a large opening, and you transfer the scraped cells with the plastic Pasteur pipette into this 15 milliliter centrifuge tube. Then you close the tube, and you spin it for 5 minutes at rather low speed. So these are 1,000 rounds per minute. And the centrifuge, of course, needs to be pre-cooled at 4 degrees. Balance the tubes, close the centrifuge, and turn it on. 1,000 RPM, set time, 4 degrees, 5 minutes, and off you go. When the run is ended, you come again, collect your samples, go back to your working place, and there you see this little pellet here. This pellet down here are the nuclei, are unbroken cells, cell debris. Yeah. Excuse me, we have to go back to the video because this was unfortunately a mistake I made. So we have to come forward to the step here. Here you can see this pellet. Now, everything which is above here is the post-nuclear supernatant, and this is what we call the nuclear pellet. Now, this nuclear pellet is discarded in our protocol now for purifying endocytic vesicles, carriers, late endosomes, lysosomes. But when you want to work with the nuclei, excuse me, this is your starting fraction, and you further you throw away the post-nuclear supernatant, and you use this nuclear fraction for further purification, also via density gradient centrifugation, detergent solubilization, and so on and so forth. Now we proceed with the video. So he aspirates the supernatant. Sorry, this is the cell pellet. This is the cell pellet we are in the first centrifugation. So I have to correct me. This is the cell pellet that we are going to homogenize now. We take away the supernatant that we have washed off the cells. Sorry, I was one step too far already. Now he adds the homogenization buffer, which is basically something like a PBS buffer, but it contains phosphatase inhibitors, protease inhibitors, a bit of a different salt concentration that is given in the protocol. You add 2 milliliters of this, and you close the tube. Make a balance tube, and then you centrifuge that for 10 minutes at 2,500 rounds per minute. So the first centrifugation was to pellet the cells without breaking them. The second centrifugation is now washing these cells already in the homogenization buffer in order to rebuffer them, that they are in the right buffer then. And now he wants to generate a kind of strong cell pellet, sets the speed at 2,500. Now the syringe is going to prepare, and he fills it a bit with H buffer to cool it also with the buffer, and to get rid of air bubbles, to blow out all the air bubbles that are trapped in the needle. So this is a 1 milliliter syringe, and he takes now this needle here. Takes off the protection, opens the tube with the homogenization buffer, fills the syringe partially with it, and with the finger he tips on the syringe that all the air bubbles go up here, and they collect here, very similar like the nurses in the hospitals are doing it before you get an injection, or by the doctors. And then you blow out the material in a piece of tissue, and then all the air bubbles are gone, and the syringe is filled with the homogenization buffer and is prepared. The homogenization buffer is only in the needle. But what we want to avoid by this procedure, air bubbles generation. And air bubble generation is always bad. So again, carefully aspirate the supernatant and leave the pellet as untouched and intact as possible. Then you take a blue tip pipette and put 200 microliters volume on it. Respond the cells gently in this by just blowing from top on the pellet. And then they start dissolving, and then you suck them in and out. Remember, we do not want to break them. We just want to have a milky, uniformly milky solution, that all the cells are in solution, and that we can start now disrupting them with the needle approach. This is a 25G needle attached to this 1 milliliter syringe. And then you aspirate. And then you blow in by blowing on the tube wall that you avoid generating air bubbles as good as possible. And the devices you can use for that are simple, as you see. These are 15 milliliter tubes or 50 milliliter tubes, depending on what your volume is and the volume of the syringe. Syringes, needles, all things that you find in every lab. You don't need special instrumentation for that. We will talk about special instrumentation a bit later. But one of the principles in science is simple is magic. Use easy things first. So now this sample has received several strokes. He takes the needle out, closes the tube, and now we pellet the nuclei out, what I wrongly explained before. Now we pellet the nuclei out, and you see we increase the speed of the centrifugation now to 3,000 rounds per minute. And we do that for 10 minutes. And here you take a fraction of the post-nuclear supernatant, an aliquot into for further analysis when you do gel electrophoresis or whatever you do, or you recover the nuclear fraction. Here we take the supernatant then for further purification via gradient centrifugation, for instance. And what I'm giving you here, these are, for instance, the numbers, so the 100 microliter the students need in our practical course, for instance, for the analysis they are doing further downstream. But this is a special protocol that we designed for this course. Now, this very nicely demonstrated you this step of the homogenization of the getting the cells off. Now we make the first fraction, and that is a simple fractionation. Just 100,000 G centrifugation in an ultra centrifuge, where we separate from this post-nuclear supernatant everything which is membrane bound, total membrane fractions in the cell, versus what is not membrane bound, by definition, the cytosol. Put that in a ultracentrifuge. So these are so-called tabletop ultracentrifuges. They are fairly small. We will talk about centrifugation a bit later. And so the students generate, for instance, in our practical course, their first fractionation into cytosol and total membranes by a centrifugation in 100,000 G. So this is 100,000 times the Earth acceleration for 1 hour at 4 degrees Celsius. I will raise a few words of caution for centrifugation a bit later. Now we come here back, remove the rotor from the spindle after the centrifugation, carefully transfer it to the bench and open the rotor, take out the tubes, and then we proceed with the experiments. So with this, I will close this video here. And introduce you to a method which is called nitrogen decompression. So do you have any questions so far to what I told you?
No.
Good. Nitrogen decompression helps you out when you have large volumes of cell solutions, or you have large volumes of cells that are growing in cell factories. So what is that? I give you an example. When you use normal plastic dishes or flasks for growing cells, they grow up to a certain confluency. And when they start touching each other, they have something like contact inhibition, epithelial cells, for instance, and also fibroblasts start then slowly growing on top of each other, but they slow down the growth. It's a natural regulation, like in an organism would take place. Now, when you take HeLa cells, which are kind of epithelial cells from a cervical cancer isolated originally, and you grow them in bottles that roll all the time, then the HeLa cells will not attach to the plastic where they normally attach to the plastic, and they grow, they divide, they make mitosis, and they grow, but they will grow in solution. And after a short time, they will adapt to the growing in solution, and they will start dividing in solution. And by this trick that you roll the bottles in the incubator on special electric devices, on rollers, you can produce huge amount of cells, and that is what we call cell factories. The same thing applies, for example, when you grow large amounts of lymphocytes and expand them in medium, they all grow in suspension. And as we have learned before, everything that grows in suspension and is roundish is much more difficult to purify. So how can we help out here? And you can also imagine with a 1 milliliter syringe or a 10 milliliter syringe, you have a certain capacity, but you cannot not over exceed that. So with the larger the volumes are, you cannot use 300 syringes, for example, or 500. Therefore, the solution to that problem is a stainless steel container where you can apply a pressure. And this stainless steel container has a valve. In this stainless steel container, you pour in your cell solution. Then you close that vessel, and it's closed. And you see there's a tubing coming in, and that comes from a nitrogen source. And then you pump nitrogen under high pressure into this vessel. And what happens is that when you pump this nitrogen under high pressure into the cell solution, the nitrogen makes a phase transition and diffuses into the cells. And when you all of a sudden it becomes liquid basically, and when all of a sudden release the pressure, it makes a phase transition again and goes into the gas phase, forms bubbles, and these bubbles disrupt all cellular contents in a very controlled way and only once. And the cells release their content, and you can collect it here on the bottom. Now, basically, this is something like a diving accident. You know, when a diver goes deep, the nitrogen goes into the liquid phase due to the surrounding pressure. And when the diver dives up too rapidly to the surface, and the pressure is released too rapidly, this phase transition is taking place again, and the nitrogen, like in the nitrogen case here, the gas goes into bubbles and disrupts the vasculature, for instance. Yeah, that is why these divers are bleeding, why the spleen is damaged, why the microvessels are damaged, the capillaries are damaged, and why you have to put them into a pressure chamber and slowly, over many hours and days, release the pressure. So this is basically the diving accident of the cells. These instruments are cheap. You find them in many institutes. They are not expensive. And as you can see here, they exist in different sizes, from several hundred milliliters up to half a liter or so. So it's upscalable. So this is maybe something that is very helpful. Here you see this procedure in more detail, how that is poured into that vessel. Now this is now our cell solution, yeah, that we collected directly from the incubator. The advantage is you could even do that in medium. Yeah, you would not even have to change the medium, which could be for example, of interest when you want to make a proteomic snapshot of cells under certain growth condition or starvation conditions, and you would like to capture in minute frames the signal transduction pathways or things like that. Keep that in mind. Now here this microvessel is closed, it's connected, and then when it is opened, the valve, you can collect the suspension of disrupted cells underneath. It has several advantages. It sounds brutal, but it is a very gentle method without chemical and physical stress. Why? There is no heat damage due to friction. You know, the more often you apply a friction to a syringe or to a plunger, you generate heat. There is no heat damage due to the friction. There is no oxidation. Any suspending medium can be used. Each cell is exposed only once, and the product is uniform, and it's easy to apply. Now we come to the point where we talk about organelle separation and density gradients. And I think this is the point where we could introduce a short break. We could make a coffee break, let's say, so 5 minutes or so. It's now 20 past 10 and we reconvene at 25 past 10, okay?
Okay, thank you.
Okay.
So we are back again, and we proceed with our lecture plan after extensively discussing homogenization. We have finally received our post-nuclear supernatant, or the first cell fraction, which is everything which is soluble in the cytoplasm or everything which is membrane associated. The next separation step in all protocols, regarding less what comes here, density shifts, free flow, immune isolation, fluorescence activated organelle sorting, the next basic cornerstone is always density gradient centrifugation. What is that? We will explain in the following. Now, during centrifugation, organelles either sediment or float due to their biophysical properties, which are defined by proteins and lipid contents, and until they reach in such a gradient a position that we call isopycnic within the density gradient. And the density of an organelle depends mostly on its physical properties, which are protein-lipid ratio and shape and size. So that means an elongated organelle behaves differently than a roundish organelle. A large organelle behaves differently. It's heavier than a small organelle. Example given, a synaptic vesicle is small, has a small diameter. An endosome has a small diameter up to 100, 300 nanometers and is round. A mitochondrion can be very largely extended, and they are tubular networks, and they are heavy organelles, so they would sediment more rapidly. And the flow of the work is what we have discussed so far, the cells or tissues that get homogenized, and then we apply sucrose gradients and separate organelles. It does not necessarily have to be sucrose. It can be any other separation medium. Ficol, for example, Bercol. These are synthetic separation media that are used, for instance, for purifying lymphocytes from patients out of blood or bufficoats. Nicotens, metrizamide. So there are many of them. Sucrose you find in every lab. It's cheap and it fulfills everything, all criteria that we will need. When you design a centrifugation protocol, it is important to keep some important precautions in mind. The more dense a biological structure is, the faster it sediments in the centrifugal field. That is logic. The more massive a biological particle is, the faster it moves. Also clear. The denser the buffer system, the biological buffer system is, the slower the particle will move. So when you have a high sucrose density, high sucrose concentration, the particles move slower. The larger the frictional coefficient is, the slower a particle will move, and the greater the centrifugal force is, the faster the particle sediments. Is also logic, but one has to keep that in mind in order to understand the principle. So the sedimentation rate of a given particle will be zero when the density of the particle and the surrounding medium are equal. This is this isopycnic position in a gradient. There are different principles of centrifugation. The first one is shown here. It's the so-called fixed angle centrifugation. The rotor has inlets where the tubes are inserted, which are tilted in a certain position in a certain degree. Such chambers are cooled. So this is an important thing because the rotation at that high speed would cause a tremendous heating and friction of the heating up of the metal rotor and then of your sample. And you can only achieve these high speed centrifugations that are required when you have a vacuum system. So once you close this centrifuge that has been pre-cooled, you have inserted the rotor on the spindle, you have this motor on the bottom, then A vacuum is generated because you want to pump out all air in order to reduce the friction that you can reach this high speed. And then the rotor starts rotating, and you will collect the samples here, as you can see here in a fixed angle rotor. These fixed angle rotors are useful for spinning out cells, bacteria pellet, cells pellet in large amounts. For subcellular fractionation, we rather use swinging arm rotors, swinging arm centrifugation. This is basically where most of the cell fractionations are done. And these are swimming arm rotors where this connects to the spindle. And during the centrifugation, these arms let the tubes that are closed rotate out into a horizontal position. And the centrifugal force, in contrast to here, where it goes on the side, it goes here to the bottom of this metal bucket where the tube is inserted. And so they are free to swing out. When you look at a cell homogenate before centrifugation, everything is easily distributed. After centrifugation, you have a straight pellet with the more dense components. So in our case, in the practical course for the cell biology students in Innsbruck, they prepared two fractions, a cytosolic fraction and a total membrane fraction for the further analysis. And this is exactly what is shown here. This would be the total membrane fraction out of the postnuclear supernatant, and this would be the cytosolic fraction. Now, centrifuges are expensive instruments. They cost as much as a Porsche or Ferrari cost, and therefore you have to care about them, you have to maintain them. And they can also be dangerous instruments because they generate tremendous forces, 100,000 times the Earth acceleration in a vacuum system. You can imagine what happens when the vacuum breaks or when rotors are not balanced. You destroy either the centrifuge or the room where the centrifuge is housed. Therefore, you have to take care. You have to take care about corrosion and degradation of the biological buffer system used within the rotors, because they should not oxidize the metal of the rotor or contaminate the interior and exterior of the centrifuge via spillage. This may seriously affect the lifetime of the equipment because they are in the bottom of the centrifuge, there are sensors that measure the speed. You can destroy these sensors, or when the spindle is dirty, it does not arrest in the correct position, and all these things in these fine mechanics and electronics that are built in can be destroyed. And these centrifuges are really expensive, and a rotor starts around 15,000 to 20,000 euro at the cheapest. Another important point is the proper balancing of the centrifuge tubes. This is not only important in terms of the safety, but it might also cause vibration-induced damage to the rotor itself and the drive shaft of the centrifuge. Thus, proper handling and care, as well as the regular maintenance of both the centrifuges as well as the rotors, is important. So they have to undergo a yearly service, and the maintenance technicians have to come to the institutes and look if the rotors are still usable. Now, when you use alkaline solutions, for example, they can cause corrosion of the metal, acidic buffers, aggressive detergents, or salt concentrations. So the rotors should be thoroughly washed with distilled water, deionized water after every run. For overnight storage, rotors should be first left upside down to drain excess liquid at room temperature, and then you turn them around and position them in a safe and dry place. Be careful with the swinging bucket rotors that you avoid damage. You should dry them with a tissue paper and remove all the biological buffers, and wash it and rinse it with water. And what is also important is many centrifuges already have an electronic record keeping, but usually you find calendars or books next to centrifuges where you sign in how long the centrifugation time was, how many runs you did, The centrifuge records it, of course, and when a certain run limit has been crossed, so to say, and you have too many runs, then the maintenance is called automatically. But when you find the centrifuge in a mess, when you go there, you usually are very angry, and then you look at the calendar, who was the idiot that who was using the centrifuge before you, and also bookkeeping and the calendar planning is advisable, because you cannot just go to the centrifuge and spin for 12 hours when it's the only centrifuge at the institute. You have to book it in advance. Mechanical stress can be a problem, therefore the loads have to be balanced, and you balance them on a microbalance, drop-wise with the same liquid. Be careful that you use the right plastic tubes that are suitable for this kind of rotor and for the centrifugation speed. When you discover that rotors are corroded and you see corrosion, that is very important to immediately call the technical service, because this can weaken the strength of a rotor, and the rotors can then fail. Now, differential centrifugation is then what we use, and I will show you in the following then a video also, because it is a very easy principle. You start with low speed centrifugation, increase the speed to medium speed, high speed, or very high speed. And in the cell homogenate that you start with, you have whole cells, nuclei, cytoskeleton fractions, and that you spin out with this low speed centrifugation, like we did this post-nuclear supernatant. Then you can increase the speed and spin out everything which is in this more soluble fraction, and you retrieve mitochondria, large lysosomes by oxysomes, for example. You increase further the speed and you pull out microsomes. So these is vesiculated ER or other small vesicles. And then what is remaining here in the supernatant, it contains proteasome, macromolecules, ribosomes, viruses, everything which is fairly large machine in the cell. You can also spin that out. These are density gradients where you can take the lysosome fractions. Then you have you load, for example, the post-nuclear supernatant down here in 40% sucrose. You overlay that with 35% and 25%. You can use normal water or heavy water. Some protocols use deuterated water, some use normal water. And then you usually see these interfaces. Yeah, and these interfaces you can mark with a pen, with a water insoluble pen. Once you have generated this gradient, so these step gradients that you overlay, you have also machines that can generate gradients, but it is also very easy to generate your gradients again yourself with a pipette, where you carefully overlay these solutions of different density. And then down here are the more heavy membranes like Golgi and ER. Then you have the endosomal carrier vesicles, and the very light fractions contain later endosomes and lysosomes. Now, how you load such a step gradient is shown here. So my coworker is loading here the step gradient. Here are the different sucrose solutions that are applied in this gradient. They're all kept on ice. And every interface you mark with this pen, with this insoluble pen marker. Be sure that you have an insoluble pen marker, because you will generate condensed water, and then they will swim away. And at that interfaces, you can then take away directly with a pipette the entire interface, and you will see a milky interface where these organelles accumulate after centrifugation. This is such a swing out rotor on the bench. This is the bucket where I loaded, and you see each of the buckets has a lid. And what is important is that these lids are numbered, and the buckets are numbered, because they are balanced, that they have equal weight and they have a position here in the rotor. So bucket number 1 should go in position number 1 with lid number 1. Counterbalanced on the other side is then bucket number 4 with lid number 4 in position number 4. And when you run, for example, only one gradient with the biological sample and not 2 gradients that you can balance, you have to counterbalance the tube on the other side also with a gradient. Why? You should not just take water to the same volume. You need the same weight and you need the same distribution of the physical weight. And therefore, you have to generate basically an avatar gradient that you load on the other side. And then you go to the balance room where the balances stand, the electronic ones, and with little drops, you pipette in the buckets the weight of both gradients until they reach an equal weight. Then you close them and you centrifuge. This is important because we are centrifuging here with 200,000 G, 100,000 G. So these are tremendous speeds. And then the sucrose solutions distribute differently. Afterwards, when you take out your tube, this is after the centrifugation, you see where we have placed these marks with the insoluble marker pen. And you see these milky bands appearing here. And these are the different organelle fractions, as simple as that you can take off and you can analyze them. Now I'm showing you a video explaining this differential centrifugation. Please give me a sign in the chat when you don't hear the tone.
In subcellular fractionation, cells are disrupted and their components are isolated into relatively homogeneous fractions. The first step in subcellular fractionation is breaking the plasma membrane. A high-speed blender can break the plasma membrane and endoplasmic reticulum into small fragments, while leaving nuclei, mitochondria, and other organelles intact. The suspension of broken cells is called a homogenate, or a lysate, and is now separated into like components by a series of centrifugations. An ultracentrifuge can rotate samples at very high speeds, over 100,000 RPM, to produce forces up to 500,000 times greater than gravity. However, the first spin is usually set at a low rotation speed, producing a centrifugal force of about 800 times the force of gravity for 10 minutes. During this spin, the largest and densest materials sediment first, forming a pellet at the bottom of the tube. This pellet will contain unbroken cells and nuclei. Thus, an enriched fraction of nuclei can be recovered from the pellet, while the other cell components are suspended in the remaining solution, called the supernatant. The supernatant is now centrifuged at a higher speed, producing a force of 15,000 times the force of gravity for 10 minutes. To sediment mitochondria, lysosomes, and peroxisomes. The supernatant contains smaller and less dense components. Recentrifugation of the supernatant at an even higher speed and duration sediments fragments of the plasma membrane and the endoplasmic reticulum. A fourth centrifugation at a still higher speed sediments ribosomes, leaving only the soluble portion of the cytoplasm, the cytosol, in the supernatant. This series of spins at higher and higher speeds and durations is called differential centrifugation. In differential centrifugation, subcellular components are separated by their size and density, with larger and denser components sedimenting faster than smaller and less dense components. The fractions obtained from differential centrifugation correspond to enriched, but still not pure, organelle preparations. For instance, some smaller particles will be found in all pellets, considering some of these particles are already present at the bottom of the tube in the original homogenate. A greater degree of purification can be achieved by resuspending the pellets in solution and then performing density gradient centrifugation, in which the sample is made to sediment through a gradient of a dense substance, such as sucrose. At the top of the gradient, the sucrose concentration is lower than at the bottom of the gradient. There are two main types of density gradient centrifugation: velocity centrifugation and equilibrium centrifugation. In velocity centrifugation, the sample is added carefully to the top of the tube in a narrow zone. The sucrose gradient below the sample serves to prevent the convective mixing of the solutions and keeps the particles in tight zones during centrifugation. Particles of different sizes sediment through the gradient at different rates, moving as discrete bands. In this technique, the density of sucrose at the bottom of the tube is less than the density of the organelles, so the centrifugation is stopped before the particles can form a pellet at the bottom of the tube. Following centrifugation, the different zones in the tube can be isolated in separate collection tubes. A hole formed in the bottom of the tube allows the collection of the different fractions. The fast sedimenting particles emerge first. The slow sedimenting particles emerge later. The other type of density gradient centrifugation is equilibrium centrifugation. In this technique, the concentrations of solutes in the gradient are very high, such that the density of the gradient at the highest point is higher than the density of the particles of interest. Organelles with higher densities are defined as having greater mass-to-volume ratios. During centrifugation, the particles move to the location at which their own density matches the density of the surrounding solution, and this is where they remain. Although larger particles may travel faster to their equilibrium locations, the spin is kept going long enough for all the particles to reach equilibrium. Thus, the final state of the tube depends only on the buoyant density of the particles. Equilibrium centrifugations are useful in separating different types of membranes from one another and are sufficiently sensitive to separate macromolecules that are labeled with different isotopes. A classic example is the analysis of DNA replication by separating DNA molecules containing heavy and light isotopes of nitrogen, using equilibrium centrifugation in cesium chloride gradients. The light and heavy DNA come to equilibrium at different locations along the tube. A hybrid DNA with one light and one heavy strand bands in the middle.
So I think this has summarized very nicely everything what we were discussing before and rounds up the picture in a way for you that you understand that homogenization and centrifugation, differential centrifugation, velocity centrifugation, and so on and so forth are the basic building blocks. And I give you now a couple of examples what you can reveal with this simple combination of techniques already. So for instance, endosomes are not only compartments where receptors are degraded, but they are also compartments where receptors that are activated at the plasma membrane travel into the cell and meet other signaling molecules and complexes. And this is the reason, for instance, why we in cancer research or in research of rare diseases are interested in these organelles. Not only that they very often harbor mutations for certain diseases, but they are also involved in sensing the nutrient environment of a cell. They are required to change their position in the cell when cancer cells start to migrate and so on and so forth. So that's the reason, for instance, why we are using these approaches in our research. Now, we focus now on the separation, for instance, of early endosomes from late endosomes using this subcellular fractionation technique. What I'm showing you here is a two-dimensional matrix. It's a technique that allows to visualize protein content. This is basically a two-dimensional difference electrophoresis, DIGES it's called. And what you do is you take your fractions, for example, the PNS fraction, the early endosomal fraction, or the late endosomal fractions, and you use different fluorescent dyes, like the Psi dyes, a blue one for the PNS, a green one for early endosomes, and a red one for late endosomes. Then you mix the samples, that you have purified before, you mix them all into one sample, you treat them with urea, and you make an isoelectric focusing in 2 dimensions, a 2-dimensional gel electrophoresis. So in the first dimension, from left to right, you do the isoelectric focusing that you do in tube gels. Then you take these tube gels and put them on another gel, and to separate these fractions that you have purified and the starting fraction according to the molecular weight. So the first separation is according to isoelectric point, and the second separation is according to molecular weight. And the fluorescent labeling tells you which protein with this unique isoelectric point and isoelectric point at molecular weight comes from. In that case, this green protein is an early endosomal protein. The red only ones are late endosomal proteins, and the blue only ones are in the PNS, in the post-nuclear supernatant. Those proteins which show a mixture of color are either proteins that appear in all 3 fractions, like this spot here, this is actin, for instance. When they are yellow, they are only late and early endosomes, and so on. And that you can read out with scanners, with fluorescent scanners, and can quantify it. And with these maps, you can separate thousands of proteins. And what is useful is you can visualize and you see how pure your sample is. And you can calculate enrichments like a 300 times 220, 305 proteins in this sample, for instance, were up to 120-fold times enriched in late endosomal fractions. Around 300 proteins were up to 25-fold enriched only in early endosomal fractions, and so on and so forth. So with these methods are quite useful to visualize. In the routine proteomics flow, we do not use that anymore because we take the purified fractions and put them into our advanced mass spectrometers, and we can use other labeling technologies that are compatible directly with mass spectrometry. Now, such a protocol then, like we have seen it, typically looks like this. All these building blocks, you know, how you prepare the post-nuclear supernatant. You take away the nuclear fraction, then you can apply different gradients where you can purify different organelles. Even with the cytosol and the membrane fraction, you can do a lot of experiments already. For instance, when you're hunting a certain protein and it is associated with the membrane fraction, you would like to know, is this transmembrane protein an integral membrane protein, or is it a peripherally attached membrane protein that is also recovered there? Simple biochemical trick, you can do a sodium carbonate extraction at high pH, which I will show you. You can do marker analysis for enzymes, and you can combine that with chromatography, 2D gel electrophoresis, and mass spec analysis. But with this simple extraction with sodium carbonate at high pH, you can discriminate between peripheral membrane proteins, which are extracted by the method, and integral membrane proteins which cannot be extracted. That could be already in a simple experimental setup an important question that your supervisor in the lab wants to have answered, and you have a solution for that. 2D gel electrophoresis again helps to visualize this complexity. And when you combine the left image and the right image, each dot is a protein from this membrane fraction. From 100,000 G supernatant after carbonate extraction, you see here the extractable peripheral membrane proteins. From the 100,000 G pellet after sodium carbonate extraction, you see here the integral membrane proteins. All these spots you can then verify by mass spectrometry, isoelectric focusing again from left to right, separation according to molecular weight from top to the bottom. So you see how powerful these little laboratory tricks already are. And of course, what you need are specific markers to follow this fractionation procedure. And the advantage, for example, when you work with the endosomal pathway is that you can introduce markers from outside. Liquid markers, for example, that are taken up by cellular drinking, by pinocytosis, macropinocytosis, endocytosis, like enzymes like horseradish peroxidase that with a very simple biochemical trick gives you a brownish precipitate that you can follow then easily with spectrometry methods or microscopy methods with everything, where in the endocytic pathway, you can take endogenous proteins for Western blotting, for instance, which are specific for these organelle boundaries, like for the very early endosomes, RAP11, RAP4, the transferrin receptor, which all recycle for these tubular recycling endosomes to the plasma membrane continuously. RAP5 as a bit of a later early endosomal marker, multivesicular body markers for the late endosomes, RAP7 for the lysosomes, beta-hexosaminidase as an enzyme that you can read out enzymatically. So meaning you can use that for looking how pure your fraction is. You can define a gradient in which fraction you find which marker, and you see how good your separation is. And you can use that, for example, the internalization protocol to internalize a marker, because we can internalize here a marker. Now, how do you do that? You take out your cells from the incubator, and you take the medium off, and you add the solution with that marker to the cells. Now the cells, you put them back in the incubator, and the marker is then continuously internalized. And you can use the time of internalization of knowing where your marker reaches which compartment. So when you put on ice the cells and you put this horseradish peroxidase or the marker on the surface, it will bind to the surface, but it will not internalize at 4 degrees. Only when you put the cells then back in the medium where this marker has bound to the plasma membrane and you put it back to the incubator at 37 degrees, protein transport is turned on again, the enzymes and the motor proteins become active, and within a few minutes, these vesicles are formed from the plasma membrane, which are internalized, and they fuse with this pre-existing compartment, the early endosome. And you can chase that further. You can take out the cells now and put them on ice again and fractionate them. Then all the vesicles that you fractionate out of them that carry that markers are by the time definition early endosomes. And you can carry that further because you know after 15 minutes you reach the multivesicular bodies, and after half an hour, for instance, you reach the late endosome and later the lysosome. What you also know is the transport step from early endosomes towards this late endocytic compartment, which typically sits in the perinuclear region of the cell, is dependent on microtubule transport, on the presence of intact microtubules and on the presence of microtubule motors. So inward transport is dyne independent. Outward transport is kinase independent. So when you would disrupt the microtubules, you would still bud from that compartment and generate this vesicular entity, but this will never reach the late endosome and the lysosome. So with that simple trick, you can enrich for this compartment. And such protocols look like that. So you internalize for early endosomes 5 minutes, for carrier vesicles 10 minutes, plus 30 minutes chase upon microtubule depolymerization. And this microtubule polymerization is achieved by nocodazole. Yeah, this solubilizes the microtubules. They can be washed out again when you want, and they polymerize again. And the late endosomes you reach with a 10 minutes internalization and a chase of 30 minutes. So very simple protocols, but very powerful. And what you also need is quality control. You always have to establish something like a balance sheet. What do you put in? Which marker you find where? How much of protein have you recovered there? And this gives you the specific activity and shows you how well you can separate. We will not go in very much detail, but this is what these balance sheets look like. You collect the fractions and you number them, and you have the post-nuclear supernatant fraction. You collect the late endosomes, the early endosomes, and you see they come from different positions from that gradient. Position 7 to 9, position 13 to 15, and the postnuclear supernatant. You write down the volumes that you have, you measure the protein amount which is in there. This gives you the protein concentration, volume, and amount. And then you measure the HRP activity with a standard curve, and then you can retrieve the amounts and the specific activity and the enrichment. And this gives you a feeling about the yield. This is important when you use that routinely, that you record it, because it's a kind of quality control. These recordings always have to be made carefully with all experiments that you're doing in your lab book, because your supervisors can only help you finding mistakes or finding out why experiments don't work anymore and why they should work again, when they can look in your lab books at your careful notes and pinpoint immediately what must have gone wrong. Like the passage number of the cells, for instance, that I told you before. Now, these gradients can be analyzed with markers by Western blot. Here you see a sucrose gradient from 10% to 40%. The fractions that have been collected are numbered on the bottom. And we are using here late endosomal markers, early recycling endosomal markers, and other specific organelle markers. And you see that you very nicely can separate these markers from early and late endosomes from each other. That means within that boxed area, you have a fairly pure gradient fraction already that is very useful for your further analysis. What you can also do is you can analyze that with enzyme assays or other markers. And there you see that, for example, here the late endosomes very nicely separate away. But in this area where you would have the early endosomes, you have, of course, a lot of plasma membrane also present. Early endosomes come from the plasma membrane, so they should have similar biophysical properties and protein lipid composition. And you still will have Golgi fractions. So what I'm trying to say is that these methods are good, but they are not perfect. And sometimes you will need higher resolution. Impurities, we call that. Cross-contaminating organelles. Here you see a nuclear fraction that we prepared in the lab, and a colleague of us at the histology department has done a scanning electron microscopy. Here you see a nucleus in higher magnification, and you see these remnants of organelle pieces sticking to the nucleus. Now, this is a typical contamination that we record, and these are parts, bits and pieces of the endoplasmic reticulum, for instance, which are marked here with arrows. So this shows you where the enemy stands. And you can then combine this sophisticated analysis with organelle proteomics analysis to learn about the protein composition of the proteome. And therefore, we are generating these organelle proteome maps, or maps of large protein complexes. And over time and signal transduction and disease processes and circumstances, we can then learn how dynamically these proteins shift between these different compartments, and when signal transduction is turned on one part of the cell or turned off on the other part of the cell. This I will skip, because this we don't need yet. And we come to the next chapter, and this is density shifts. Now, everything what we achieved so far are density gradients, and now I would like to show you a fine-tuning of these density gradients. For instance, you remember the problem we had with the early endosomes, that within the gradient, these early endosomes were recovered in a fraction where plasma membrane is, and in a fraction where most likely Golgi or a bit of ER is. So how could you solve that part of the gradient, that fraction? Now, you could apply density shifts. You can load these endosomes with nanoparticles, colloidal gold particles bound to a ligand, for example, transferrin, or bound to an antibody that is internalized. Then you internalize them into the early endosome, for instance, and then you fractionate yourself, homogenize your cells, fractionate themselves, you load the gradients again. You take the fraction where the early endosomes are in, but they are now in a different fraction, because after the separation through the centrifugation, this fraction will change its behavior in the gradient through the content of the colloidal gold of the nanoparticles and will move to a different position. And you will get rid, for instance, of the plasma membrane and the ER. The same can be applied with HRP, for example. HRP you can bind to a ligand like transferrin and separate this by centrifugation after you have treated your sample with diaminobenzidine. This is a very simple chemical reaction that cross-links the luminal proteins, this HRP, and then this HRP containing vesicles through this cross-linking form a precipitate. And this precipitate changes significantly the behavior of such vesicles on a gradient. Just another example. Now we come to a more sophisticated method, and that is the method of free flow electrophoresis. And this deserves now some attention from us. Free flow electrophoresis is a preparative separation tool where you can enrich proteins, protein complexes, and organelles. You have to pre-fractionate your samples, and you can load these complex mixtures, and you can use pH gradients that are performed in a flow. And the principle I will explain you, and that has been very successfully used for the separation of large protein complexes already and organelles. And the samples you can apply provide in buffer or salt solutions that should not have to a high salt content. And the reason is shown in the next field, because you apply horizontally a very strong electric field. Now, the principle of free flow electrophoresis is that you have a plate where you can flow electrolyte buffers. And this is called then zone electrophoresis. You apply the sample in a segmented electrolyte in a flow that goes from here to here. You apply it here, and you apply horizontally an electric field. And due to the isoelectric behavior of a protein, in that electric field, the protein can be deflected. The same is true for an organelle, because an organelle has a netto charge. All proteins and all lipids together contribute to a certain charge, and that will cause a certain behavior in a free flow electrophoresis, a deviation from other organelles. And at the end of that field here, you can recover the sample in 96 well plates, for instance, and you can fractionate up to 96 fractions. You can form a segmented electrolyte, a uniform electrolyte, and several segmented electrolytes. And by that principle, you have a large variety that you can apply. Now, this was originally discovered by a researcher at the Max Planck Institute, and was then taken up by large companies who provide proteomic devices. And in the following, I will show you a video where this principle is explained in detail. Well, this was free flow electrophoresis, a very, very powerful tool, an expensive tool, I have to say. You find it basically in every large mass spec unit or proteomics unit. In Vienna, I think there are two instruments mounted, either for enriching protein complexes or organelles. But as I said, in this second part of the lecture, you learn specialized techniques that you hear about them. But these are all techniques that require special equipment. And what I am trying to teach you also are techniques that you can perform yourself. The next technique is a technique where you don't need very much equipment. It's called immune isolation. And this is going to be a very interesting technology. Now, what is immune isolation? Here, you separate organelles with antibodies according to their antigenic properties rather than their physical properties. However, you combine it most efficiently with homogenization, transient gradient centrifugation as means of prefractionation. And those techniques you bring in when the conventional differential centrifugation approaches do not work anymore and you need higher resolution. In most of the cases, this is not required. However, it might be that your supervisor tortures you and tells you, no, your fraction is not pure enough. We still have contaminations in there. I am not sure if my protein of interest or my protein complex of interest is really on organelle A or B, or it's brought in by a third contamination. Therefore, you take the antigenic properties of these organelles and you pull them out with solid supports that are linked and coupled to antibodies. Basically, it's something like immunoprecipitation that you might have performed. There's a big difference. Immunoprecipitation, where you immunoprecipitate proteins and proteins that are associated with your protein complexes, are run under denaturing conditions in the presence of detergent. Because you have to solubilize the membranes, you have to get access to the protein complexes, and these are denaturing conditions. You pull them down with antibodies that are hooked to beads, or to fibers, or magnetic beads, and you pull them out under these denaturing conditions, and you analyze them on gels. What we are doing here as immune isolation are native conditions, because we want to keep the organelles intact and as native as possible. So what we need are several requirements. We need a solid support. We need a specific antibody that recognizes an epitope that is accessible on the organelle fraction. And we need a linker antibody that recognizes this antibody and hooks it to the solid support. And as you can see here in this very simplified scheme, fractions that do not contain the antigen will not be retrieved. So that means you can further select for specificity and you can further enrich the sample of interest. The experimental strategy can be a direct strategy, as shown on the left, or indirect strategy. What is the direct strategy? In the direct strategy, you have your solid support, this nanoparticle, with these three of the two antibodies. Now, this antibody that recognizes the primary antibody, so to say, binds to the Fc part of this antibody and is hooked to these beads. Thereby, you have already the correct orientation of the antigen binding site towards outwards. That means you have a very good fishing device. And these preformed beads or trees of antibodies on these beads, you mix With your gradient centrifugation enriched purified organelle fraction, you let that that bind for some time, and then you retrieve the beads out and you analyze them with the organelles bound to it. The indirect method is very similar, but has a different principle, because you add the primary antibodies first to your organelle fraction, let it bind, and then you add the beads with the secondary antibody bound to it, and you fish out everything that is bound here. Now, both methods work. It's most of the cases error and trial to find out what works best. What is crucial, and that is the same principle for the immune precipitation, or the ones of you who have done immune precipitations already, is you have to define the experimental conditions. When you have an antibody binding to an epitope, the specific binding takes place in a short time, and then this curve flattens out and you do not have more specific binding. However, the non-specific binding is going to increase over time, and at a certain moment these two curves will cross, and then you will have no specific purification anymore. At the same time, when you look at the areas underneath the curves, you increase non-specific bound material already very early, and you want to keep these contaminations here in this triangle as small as possible. So what are we usually doing when we're setting up such protocols? When we have a new antibody and a new protein or a new organelle fraction we want to purify, we have to test what works. Usually, when you keep this antibody binding reaction at 4 degrees, it's very slow. It's much slower. But much, much slower is also the non-specific binding. That's the reason why you see these rotating wheels with tubes for immune precipitations in every cold room that go for hours. In principle, you can perform the same reaction when the antibody is highly specific at room temperature, which might be beneficial for some antibodies. When they have a very high affinity, they bind very rapidly. But you have always a kind of window. Within this window, you can walk. Room temperature is not good for your protein solution, because phosphatases, proteolytic enzymes, lipolytic enzymes, they are all very active. However, it might be more beneficial for the antibody. In the cold room at 4 degrees, everything is much slower, and you have to find the right balance, how long you incubate in the cold room or how long you incubate on the bench. Usually with defined antibody pairs that have been probed for immune precipitation, for example, or for immune isolation, you find protocols in the lab that tell you how long you have to put it on the rotation wheel or the rotation tube for this immune isolation or immune precipitation to be carried out. And you might wonder why there is written 15 minutes or 2 hours or overnight. The reason is that these protocols have been established. When you take a new antibody pair, you might have to establish it again from the beginning. This just as an explanation. The antigen must, of course, fulfill certain requirements. First requirement, and most important, is that the epitope must be exclusively present on the surface of the desired compartment. It does not help when you are looking up in your antibody database in the laboratory, so which antibodies do we have against mitochondria, for instance, and you find out a mitochondrial matrix protein that has a highly specific antibody. It works like a charm for immune precipitation when you solubilize it. It works nicely for immunofluorescence, but the problem is it's an epitope that sits inside an intact mitochondrium, so it will not be helpful. So be careful about that. It must be exposed on the surface, and it must be only in mitochondria. So it must be only in early endosomes or only in post-Golgi vesicles. You need, of course, a certain density of that epitope that you can purify efficiently then. The antibodies must fulfill certain requirements. The linker antibody, for instance, it increases also the flexibility of the specific antibody and the orientation. So when you have a linker antibody, your fishing device will be more flexible, like a cantilever. Yeah. And you have a bit of a longer orientation, and you have the right orientation that the antigen binding site is really nicely facing out. The specific antibody that you are using should find the epitope that is exposed in the surface. It should have a certain KD that it works. That means a good binding affinity. And the selection of an antibody is difficult. When you use polyclonal antibodies from rabbits, for example, they should be affinity purified. You can use monoclonal antibodies, but it's not clear. It's not clear. Excuse me, I think my iPhones. Do you hear me still?
Yes.
Because my iPods gave up, and I will switch to the internal mic. So now you should hear me again, okay? Can you hear me?
Yes, we can.
Thank you. So it cannot be predicted if an antibody will work finally. And that means you have to test it. You have to make an experiment to try against the same epitope, maybe different antibodies, to find out for which purpose which antibody works best. So for example, there are antibodies against the EGF receptor, for instance, on the plasma membrane that work very nicely in Western blots. But they don't work in microscopy. There are some of them that work in immunofluorescence under certain fixation conditions, but not in electron microscopy and vice versa. There are some of them that work in immune precipitation only under denaturing conditions, and some of them work under native conditions as well. So these are all features of the antibodies that you have to find out and define yourself, if they are not already defined in your lab where you work. But this might explain to you why in certain protocols it's written to use this antibody from this supplier with this catalog number. The solid supports have also to fulfill certain criteria. The composition. Now, hydrophobic surfaces are more sticky in general because the membranes can attach. They should be basically accessible for a simple chemistry that you can attach the antibodies, and they should not aggregate themselves, because when we saw, for example, some latex beads from aggregate, and every time that we have heard today already in the cellular context, when you form an aggregate, you increase unspecific binding. The flexibility is important, that you can position the antibody by different means. And you have basically at the end of the day, you have to get them out of your solution again. So either they should be sedimentable, or you should take them out maybe by magnetic force without aggregation. These are common solid support types that you find in every laboratory supplier. The most commonly used nowadays are magnetic beads. They have a low background as advantage. They do not sediment themselves. They are monodisperse of uniform size, commercially available with a variety of antibodies already coupled to them, but they are at analytical grade. They have low capacity. Yeah, so that means they are good for running a gel or for analyzing, or maybe with one mass spec shot, but these are not preparative methods. Preparative methods are cellulose fibers. So the same principle, but in fibers, where you can hook antibodies on this fibered meshwork, and it's like a fishing net, basically. So it has a huge capacity, has a high flexibility. At low speed, it sediments, so this is also important. But they have, of course, an entrapment background, like a fishing net that you pull out things from the water, from the sea, from the lake that you don't want. You can produce them yourself. They are not commercially available, but for some purposes, they are powerful.
Or per-sheet particles are something like porcelain particles. They are more or less monodispersed, commercially available. They have a huge capacity, but the background is also higher, and they work like a charm for some antigens, but not for all. And there's a natural source, which is up here on the left, and this is fixed, chemically fixed Staphylococcus aureus cells that express protein A. Why protein A? Because protein A can bind antibodies, yeah, on the FC segment. They have very high capacity. They are monodispersed, beautifully produced by nature. They are commercially available. You need rather high speed for sedimenting them. And one should not forget, Staphylococcus aureus has a proteome itself. So this might for proteomics analysis not be the best idea. And this is what such an immune isolation of endosomes look like. So this is again a duty-cellular electrophoresis. And now, all of a sudden, we have not thousands of spots there. We have only a few hundreds of spots. And that is very, very close to the real proteome of an organelle without contaminations. So this is a technology you could think about, and you can use it basically for every organelle, and that you can pull out immune isolation of organelles. You can immune isolate cilia bodies, you can immune isolate centrosomes, you can immune isolate all kinds of organelles that you find in the cell. Keep it in mind, it's not expensive, and you can do it with basic equipment. The last methodology we're going to introduce is fluorescence-activated organelle sorting. And this uses an instrument you most likely have been introduced to you already, especially when you worked, when you were in immunology lectures during your courses and seminars, or when you worked in an immunological institute, and that is flow cytometry. Now, flow cytometry is used to sort out cell populations that are decorated with certain antibodies that have a certain color. It is as simple as that. And that can be, the same instrument can be combined with conventional subcellular fractionation techniques and high-speed organelle sorting in a FACS, in a fluorescence-activated cell sorter. And we call it fluorescence-activated organelle sorting. Very elegant technique, and you can basically use every FACS that you find in an institute, and you find many of these instruments usually. The principle is that you label these intracellular organelles, the mitochondria, the Golgi, the ER, the plasma membrane, the phagosomes, the endosomes, with fluorescence membrane dyes or fluorescently labeled ligands that you can internalize. And this allows purification due to biological properties rather than physical densities. However, you can most conveniently combine it with the homogenization that was teaching you today and the differential centrifugation technologies. This is the technical principle of such an instrument of a fluorescence activated organelle sorter or cell sorter. The sample comes in a stream, in a tubing from a reservoir. Then you have a sheet, you have a sheet flow. And this is mixed here with the buffer. And then a hydrodynamic focusing takes place in most of the instruments. There are many other principles as well, but this is enough for you to understand. This is a nozzle that vibrates. And this nozzle vibrates in a way that the stream is getting focused, is getting into that elongated part of the nozzle, and is then droplet-wise released. And this happens statistically so that in every other droplet is either a cell or organelles. Now, these stained cells or stained organelles are passing through here where we have a light source and the detector system. And you can record two different qualities of this light. One is the scattering of the light that gives you an indication of the granularity or of the shape of a cell. Yeah? You have a forward light scatter, for instance, and a horizontal light scatter. And you can record the fluorescence light in different colors. Here's just shown red. Now these droplets are generated, and the droplets that carry the fluorescence signal, they get through a computer device, when they flow between an electric field that is here at the end, they get a charge when they are released here from this nozzle. And due to that charge, when they are red, they get a charge. When they are green, they get a different charge. Then they are deflected to one or the other side. So here you can collect these deflected droplets. In the middle goes the waste that has nothing, and here go the red, and here go the green sorted cells. And the same thing you can, of course, apply for organelles. The criteria for that, that the sensitivity is obviously a major concern because organelles are small structures, much smaller than cells. And therefore, they have only a small number of dye molecules associated with them. You have to take care about the physical properties of a dye, for example, the absorption coefficient, the quantum efficiency, and you have to take care about fluorescence background from cells. Why I'm saying that? Amino acids, some of the amino acids are autofluorescent. And when you have, let's say, metabolically very active cells, sometimes they can be autofluorescents per se. Typical example given in immunofluorescence, you always include a primary antibody control, a secondary antibody control, and where there is no antibody in there, and all of a sudden you see that where there should be no fluorescence, there are some cells green. And that's the reason, because they are metabolically extremely active and they produce autofluorescence and amino acids. So for which organelles, again, endosomes you can use, but everything else you can use mitochondria. There are mitochondrial dyes, for example, distinguish rhodamine dyes. I think rhodamine 123 is it that decorates only mitochondria when they have an intact membrane potential. And you can sort those away from mitochondria that have a damaged membrane potential and analyze the one or the other fraction. We have many cell-penetrating fluorescence stains that can stain intracellular organelles. And you can engineer organelles and protein complexes with GFP, YFP, and all other kind of fusion proteins that you can use then as markers. And this shows you again as visualization such an organelle fraction. This is the starting fraction in post-nuclear supernet, and this is the enriched endosome fraction. So a very similar picture like you have with the immune isolation. Very specific. You see proteins like here that only show up in these enriched endosome fractions, which are not here. Other proteins which are much more prominent here in the post-nuclear superfraction, the starting fraction, which are completely missing here. So this shows you already how powerful the combination of these techniques is, and that's the reason why people all over the world are establishing these proteome-wide organelle maps with different procedures, with organelle profiling, with gradient centrifugation, with a systematic mapping of all protein content that you might find there, doing principal component analysis, like in genome sequencing, in deep sequencing, where you see where certain subset of genes come up under certain conditions. You can treat your cells, you can starve them, you can stimulate them for a growth factor then again, and you can study how the protein contents changes over time in certain parts of the cell. You can generate virtual organelles, virtual phagosome maps, and map all the organelles to that. You can do the same thing with lipids. This shows the organelle lipid distribution profile. You know, mass spectrometry has become the most powerful technology for lipid analysis. All subspecies can be mapped, and that can be very efficiently combined with organelle fractionation. Now, we come to the end of this course, and you will get a homework. In order that you get a mark for your registered course, you get a homework. And the homework, as in the introduction video on the YouTube channel was explained already, should be an essay on subcellular fractionation. And what is very important for you, the colleagues who have not been here today, who will watch the video on YouTube. And you who have been here today, you have the same homework. Therefore, everybody should listen very carefully what I'm saying. This essay doesn't have to be long, 4 to 5 pages at the maximum, can even be shorter. You can describe every topic you wish. You can give a summary of what I have shown you. For instance, I supplied you with literature. So typical protocols, isolation of endocytic organelles, subcellular fractionation in general, organelle proteomics, a review, subcellular fractionation, electromigration analysis, and mapping of organelles, organelle omics, a strategy to untangle the biomolecular complexity of the cell. All this literature you have received. You can use that. You can use your own literature sources. You can choose the topic. What the challenge is, you can link that also to your interests, to your work. You can select a single topic. You can give a summary of my lecture today. It is up to you. But what you have to do, and what is the requirement, is you use your own words and your own English. Don't cut, copy, paste text out of reviews. Write it in your own English. This is the most important exercise for you. I will look at it. And when it requires tuning or fine tuning, I will contact you. I will send you corrections back. No cut, copy, paste. And you can believe me, I will recognize cut, copy, paste immediately. Yeah, because I know the English students are writing in normally. I know the depth in what they can go. I'm long enough in that business. I can recognize immediately where the English comes from, how it is written. If it was written by a native speaker, by German, Italian, Russian, Croatian, I know I have all these students and co-workers in my lab. I correct always their manuscripts. We write the papers together. I know the peculiarities of each language, and I will recognize if you have written it alone or not. This online material that I supplied to you can be useful for your further laboratory work. It can be useful that you understand now and how to read such protocols. It can be useful material for your homework. And the entire stuff, once you're finished, you send to me under this email address. And when I receive that, I will answer immediately. And when you don't hear from me within 1 day, that I write back, okay, received, or fine, I got it, then there is something wrong. You know, sometimes I have an overflow in my email. I get around 150 emails a day. I spend a lot of time on the computer. Things can get lost. But my reliability for you is that when I don't write back within 1 day, then there's something wrong and give me a reminder. Yeah. Good. Now we have to agree only on one point. Until when do you think is realistic that you can do that?
Until next week.
For me, it's fine, but we need a binding date where everybody could submit it.
I would need a longer time because I have exams now.
Yeah.
So, but yeah, I don't know. So for me, it would be like somewhere in mid-July, I guess, if this is possible, because I have like 5 exams in the next 2 weeks.
Is that something we could agree on?
For me, this would work, but I don't know about the others.
Okay, it's okay.
Okay, because that we will then show up also in the YouTube channel. And then all the other colleagues, some wrote to me that they cannot, but they would like to join in the YouTube channel and do the homework as well. Do you think that's realistic? I would not postpone it too long because now you have it fresh in your mind. But 15th of July is fair enough. I have no complaint against that. So do we agree?
Works for me.
Yeah, perfect.
Good. Okay, do you have questions to me or comments?
Maybe one question. When you showed the impurities, you showed a picture of the